So we're going to get started now. I said we had 4 longer presentations. The teams have 15 minutes to present their work, and then there'll be 5 minutes for questions following. So I'm going to call the first team up, and this is a team represented today by Scott Manalis from the Koch Institute and David Weinstock, formerly of Dana-Farber Cancer Institute and now at Merck. They received one of the traditional grants in 2015 for a project called Targeting Minimal Residual Disease in Acute Leukemia. You'll hear they've made tremendous progress, including the founding of a company, Travera, which is pursuing this idea into the clinic as we speak. Gentlemen.
Great. Yes, my microphone's on. Well, I just want to start by first acknowledging Art Gelb and David Livingston. I knew David quite well, and I have to say he's one of the very few people I was truly jealous of. His intellect and his boundless enthusiasm really just blew me away. Art I did not know as well, but I felt exactly the same energy from him. And in terms of how can we continue that legacy, it reminds me after the completion of our bridge project, Scott and I were awarded Distinguished Investigator Awards from the Paul G. Allen Institute, Paul Allen, who co-founded Microsoft with Bill Gates. And soon after we received those awards, Paul actually died of lymphoma. And the organization was quite shaken. And we asked them, what can we possibly do to honor him? And they said, do the best work you possibly can. And so that's what we tried to do. So I am a physician scientist. At the time, I recognized, and I think so did everyone else in the field, that the biggest problem, especially in leukemia, was not being able to kill many leukemia cells. It was being able to cure the disease. So if a patient starts with somewhere between 1 billion and 1 trillion leukemia cells, well over 2/3 of those patients we can put into remission by killing at least 99.9% of those cells. And so patients will then enter this period when they have what we call minimal residual disease. It's very, very difficult to track that minimal residual disease. We give them additional therapy, hoping that we will cure their disease. But the large majority, especially of patients with acute myelogenous leukemia, will ultimately relapse. And they typically relapse with clones that have grown from that minimal residual disease and have different genetic mutations and different therapeutic sensitivities than the original leukemia. So that's the problem. How do we fix that problem? Well, we had the idea that if you could isolate individual cells of minimal residual disease and you could figure out what drugs they're sensitive to, then perhaps you could eradicate those cells by literally, to some extent, playing whack-a-mole. You find the cells, you give the right drug, maybe other cells grow up, you find those, you give the right drug. The major challenge, there are many potential advantages for doing that, especially because patients typically can tolerate therapies much better when they only have a tiny amount of leukemia than when they have a lot. The microenvironment may be changed in ways that actually favor the efficacy of the therapies. But the major challenge is how do you take an individual cell and figure out what it's sensitive to? And it was really through a connection that Franziska Michor made soon after the announcement for the bridge project that Scott and I were connected. So what we needed was a technology that could take live minimal residual disease cells, single cells, and be able to enrich them from a background of all of these normal cells that might be in the bone marrow and the blood. And we had strategies internally in the Weinstock lab, and I think in the larger leukemia ecosystem to be able to do that. But then the really challenging part of this was how do you take a very limited number of MRD cells and figure out what the drug sensitivity is? And it was the traditional approach for this has been to do genetic sequencing. And in certain contexts, that genetic sequencing has been extremely valuable. So patients who are treated for CML with a drug like imatinib will predictably develop mutations in their CML. You can sequence those mutations and you can figure out what's the right next drug. But when we look across the entire world of cancer, we have been much, much less successful at doing that. This is what's called the NCI MATCH study. And this was an enormous effort across scores of different institutions to find patients with mostly solid tumors, sequence their tumors, and then if there was a drug that matched the mutations that they had, give them the right drug. And what you see here is only about less than 20%, 15 to 20% of patients even had a mutation that would tell you what drug to give. And only about 1/3 of those who were actually treated actually got any benefit from that drug. So that means about 5% of people right now are getting benefit from sequencing of their tumors and trying to match it to a drug. Now, that's incredibly important for those 5% of people, but it's clearly not the answer for everyone. So I'll get into what Scott does just in this one slide, and then he'll take over with the actual technology. But I'm also trained in infectious diseases. And so we recognized a long time ago that there's a completely separate paradigm in infectious disease. Right? Someone has a bacteria, let's say they have pneumococcal pneumonia, the most common cause of community-acquired pneumonia. They come into the hospital, they cough onto a swab, that swab is then put onto this culture plate, and the pneumococcal bacteria grows all over the plate. You take these tiny little discs that have different antibiotics, penicillin and ciprofloxacin and vancomycin, and you put those discs on, and based on the size of the penumbra around it, basically the antibiotic has leached out and the bacteria can't grow there. And so what you're measuring here is the ability of the bacteria to proliferate, to grow in the setting of the drug. And this is absolutely the standard of care everywhere on Earth. It's interesting because this tells you an absolute readout of how sensitive that bacteria is to the antibiotic. It doesn't tell you if you treat the patient with a particular antibiotic that they're going to live. So you can have a penicillin-sensitive pneumococcus, but you could still die of pneumococcal pneumonia. The question was, could we at least get to that first step? Could we at least understand that the cancer cells are or are not sensitive to the different drugs in our armamentarium? And then we could apply the pressure to do the clinical trials to actually see, can we help save people's lives? So I'll hand it over to Scott.
Okay, thanks, David. So unlike bacteria, cancer cells, if you take them from the tumor, they won't proliferate ex vivo, at least in natural conditions. And so this assay that David showed you for bacteria doesn't really work. And so what we hypothesize is that the mass of the cancer cells, the actual weight of the individual cancer cells, could reveal the drug response of how the patient might respond to the drug. And so what we found out over the years is that actually this mass response would reflect drug mechanisms of action that varied widely. Like metabolic-based drugs would skew the population maybe anabolically or catabolically, which would affect the mass of the cell. A lot of drugs, and here I'm really talking about drugs that are either chemotherapies or targeted therapies. A lot of drugs will arrest cells in the cell cycle somewhere, and that would skew the population either heavy or light, depending on where they're arresting. And then other drugs would damage the structure of the cells, and then mass would kind of come out and be secreted through that. And what we found for all these different types of chemos and targeted therapies that we would test is that the response that we see in this mass typically came out in about 24 hours. And this is really important because when you take the tumor cells out of the body, they're on their way to death. It's not the environment for them. And within a couple of days, in natural conditions, they will tend to crash out. And so having this speed turns out to be very important to be able to make an assessment of the response. So how did we measure the mass of the individual cells? Well, we do it with a diving board. And so it really is a diving board, but on a smaller scale. And this thing is vibrating at a resonant frequency. And there's a fluidic channel embedded inside of it. And so we can flow cells one by one through this vibrating diving board. And when the cells flow through, the resonant frequency goes down by how much mass that cell has. Just like you can imagine if you're at the end of a diving board and flexing your legs and you jump off, there'll be a change in that frequency. That's what we measure. That tells us the mass. And we can flow cells through very rapidly, thousands of cells a minute. We can be able to survey in this way, much like a flow cytometer. And so this is actually some of the early data between David's lab and mine, where we took a cell line that has this BCR-ABL translocation. It's sensitive to imatinib. And so this is a targeted therapy that actually arrests cells early in the cell cycle. And what we see, the black point, so every point here is an individual cell that we measured its mass, and we also measured the rate at which that cell is accumulating mass. And so our control-treated cells are clustered up here in black. The color points reflect the change in mass. You can see they're skewing now towards lower mass because of the arrest, and they're not accumulating mass as much. And we were able to see these responses in hours, yet the viability, which would be the conventional readout for that, wouldn't change for a couple of days. And so this was really exciting to us that we had this kind of early window into the drug response. And then an additional benefit, because this diving board is a microfluidic system, we can take in very low input samples. And so we were able to show that down to 1,000 cells we can take into this to do the measurement. And so we moved from the cell line with David's lab to a mouse model. we took a cheek bleed for which we then fed through and measured 1,000 cells. And we plotted their mass responses here, in this case to ponatinib, and could still see a very sensitive response. And so this got us really excited about MRD and the fact that patients that are at the point of MRD, they don't have a lot of cells, cancer cells, in their bone marrow. They might only have tens of thousands of cells left at that point. But this really suggests that if we can get those cells out, we might be able to treat them and challenge them ex vivo with a panel of drugs to figure out which one works best.
And this was several years ago, I'll just add that. The 25 microliters of blood, that's much less than Elizabeth Holmes and Theranos said they needed. And we thought that was a good thing at the time.
Yes. Okay, so the bridge project that we had also had a really big impact on the trainees. And in the project that David and I had, there were two people in our labs that ended up working very closely together, Mark Murakami and Mark Stevens in my lab. And Mark Murakami, now he's an assistant professor at DFCI, and actually his work still revolves very much around MRD. My lab is collaborating with him in the context of lymphoma for this. Mark Stevens, together with a few others on the project, included David and myself, we started Travera, that Tyler mentioned, to take this to the clinic. And the last two slides, I'll just kind of give you to present day where things are. And so Travera set up what is called the CLIA lab, which means that they can take patient samples and return the results of the test to the oncologist for that patient. And so this is a lab that does a 2-day test. 1 day is for the shipping, 24-hour shipping, and then 24 hours of testing. So the patient specimen comes in, it's put into different wells. Up to 20 drugs can be tested on that patient sample. It's run through to measure this mass response as well as morphology of the cells, and then make it a simple assessment of what the mass response is for each drug, generates a report, and turns that back to the patient. Travera has, in their CLIA lab, validated over 100 FDA-approved drugs that they can now test in this facility. And just this year, Travera published a paper on a clinical study that they've done, a small cohort of patients shown here. So on this axis here are the different patients that were tested. And this y-axis here is a mass response score, just kind of an aggregate number of how much mass was changed by a drug. And in this study, Travera tested the same drug that the patient took. And so we're able to link the mass response to the outcome of the patient. And so when you see here a large number, there's a big change in mass, we would predict that the patient should also show a response. Green means the patient responds, and you see we can predict that, but there are some exceptions that we missed here. If we don't see any change in mass when we challenge these cells, we would predict that the patient would not respond to that drug, and that's mostly true when you see the red bars down here. So of the 64 patients we did, 54 of them we were able to predict their outcome to that drug. This is across many different cancers and many different drugs, and what Travera is doing now is focusing really just on 1 or 2 cancers to increase these numbers to hundreds of patients to really test the statistical significance of this assay. So just to conclude, here are the members of the team that we had at that time for our project. We are really incredibly grateful for the bridge funding because not only did it launch this project, but it really created a community between David and I that has lasted all that time until this very day that we actively are working together. So thank you very much, and we're happy to take questions.
Thank you, guys. We have time for questions. Thanks for staying on time, by the way. This is what happens when a biological engineer / physicist meets a clinical oncologist interested in minimal residual disease. Magic happens.
Sounds like a joke.
Change a light bulb, right? Yeah.
We have time for questions.
Hi, thank you so much. That was really, really cool and really interesting. I'm just wondering, to what extent do you think that minimal residual disease will actually have similar sensitivity profiles that the bulk disease that you're validating your assay in?
I think it's a spectacular question, and we don't know the answer. We've started to look, I think Mark has started to look mostly in lymphoma models at that. We have a paper that we've been working on that was part of a U54 that suggests that in BCR-ABL leukemia xenografts, that there's a subset of cells that are somewhat quiescent and have a completely different sensitivity profile, as you'd imagine, based on their cell state. But it's a really challenging question.
Michael.
Fantastic, absolutely fantastic work. For Scott and David, my question is, how much further can you take this? So clearly you're doing this with minimal residual disease, but what about, for example, circulating tumor cells in solid tumors? Can you use the small amount of circulating tumor cells to predict the response of the solid tumor to therapy?
Yeah, so it's a great question. I think it's really challenging with circulating tumor cells because the number is so low. I mean, for a lot of these cancers, it's in the tens of cells. Maybe really late stage, it might be hundreds. And our threshold really is 1,000. We can push that a little bit. But then if we wanted to test a lot of drugs, it's hard. So we would love to be able, but if people can, and there's a lot of work on this, if you can figure out how to better enrich the circulating tumor cells, maybe through leukapheresis or other types of ways, which some people have thought about, there actually are a lot of cells there if you can get them. And then I think it would be a tremendous opportunity to test those cells. Yeah.
So after a cell divides in mitosis, it's half the size. So how much is your mass actually just detected in, say, early G1 cells, or is it something else the drug is causing to reduce the mass?
Yeah, that's a great question. So mass for normal proliferating cells, cell cycle and mass are very tightly linked, as you point out. When we take these tumor cells ex vivo from a patient, they're not defined so well. They're not really in a cell cycle or proliferative state. And so those three mechanisms that I showed you, they're not like individual ones for each of those types of cells or drugs. They're kind of convolved together, and we see hints of different ones. So I think it's complicated on the ex vivo samples on exactly what the mechanisms are that are changing it, but they seem to be made up of those three different things based on the types of drugs we've tested and the responses that we've seen. But I think there's a lot to that question you're asking.
Wonderful. Thank you again. And let's thank the audience. And we'll move on to our second team. So I'd like to welcome up Michael Sima from the Koch Institute and Robert Cormack from Brigham and Women's Hospital and Greg Ekchian from Strategen Bio. This is a team that was awarded an expansion bridge grant. Again, I mentioned earlier for more clinically stage efforts. That occurred in 2017. The title of the grant was Quantitative Tumor Oxygen Measurements in Cervical Cancer Patients. And this too, just like the last example, led to the formation of a company, Strategen Bio, which indeed has moved into the clinic, extremely excitingly. So, gentlemen, take it away.
Good. Cool. Well, so thank you for the opportunity to be back. I spent a lot of time in this building and in this conference room, so I appreciate the opportunity to come back. So I have with me Michael Sima, professor here, as Tyler said, my PI when I was here at MIT, and Rob Cormack from the Brigham. And he also mentioned since then we've spun out Strategen, so that's what I spend the vast majority of my days and nights doing now. Before we really jump into it, though, I think we all want on our end recognize the loss of a really critical team member. Larissa Lee was the clinician on this project, radiation oncologist at Dana-Farber, and she passed away in 2021. And that was a big loss for the team, both personally and professionally. We've since then new members have come on and have continued the work, but just want to acknowledge Larissa's contributions and that she's been dearly missed over the last few years. So I came at this project from the engineering side. Michael and I were working on the development of oxygen sensors that you measured using MRI. And what that means is there's a signature on the MRI scanner that allows you to measure oxygen quantitatively in an absolute sense. That's not something that's possible with MRI today. And there were a lot of applications you can apply this to. So we had these little seeds that you put into the tissue. And then when you did an MRI measurement, you could extract this information. That's where our bridge project team started to organically form. So we got connected with some folks at the Brigham and Dana-Farber and started going over and making presentations about what our technology could do to actually find what indication actually made the most sense for a collaboration. As an engineer reading papers, I could come up with lots of potential ideas, but it wasn't until we actually talked to people treating patients where you realized some of these things are only discussed in papers. Some of these things are actually practical applications. So there were three criteria we really thought about. We wanted there to be existing clinical data showing that oxygen affected outcomes. We wanted MRI to already be in the workflow, And we wanted an existing invasive step, sometime where you're poking or prodding a tissue where we could actually put our sensor in. And cervical cancer radiation therapy ended up being that application. So greater than half of cervical tumors are hypoxic, and those patients have lower outcomes based on that tumor hypoxia. MRI has been rapidly increasing in its use in cervical cancer. And then lastly, there's a step where they're putting things into the tumor that we could piggyback on top of. That was important both for the clinical trial we would end up running, but also as we think ahead towards how do we make this standard of care. It's much easier if MRI is in the workflow and there's a piggyback step for insertion. So a quick overview of how cervical cancer is treated. So you have 5 weeks of external beam radiation, chemotherapy, followed by the high dose rate brachytherapy part. And this is really where we focused. Here you're putting in these small tubes that are 2 millimeter in diameter and about 1 foot long into the tissue. They're closed on the end that goes into the tissue, but they serve as channels to put radioactive seeds temporarily into the tumor to boost dose, and then you pull these out after you're done. So this is where we decided we were going to focus. We actually sat down and strategically thought about how do we get our sensors into the tissue in the least invasive fashion. And our solution was to modify a brachytherapy catheter. So we took these brachytherapy catheters that are solid, machined holes in those, placed our sensor. These stick out of the patient, so we're sealing our sensor in the tip. And now we have a vehicle to temporarily deploy our sensor in the patients. I think one thing I'll point out here is we made about 500 of these in the Koch Institute upstairs in Michael's lab, but not a single one of those met the quality standards to go into a patient. So I had I think, very informative and great learning experience of actually going and making a medical device that could go into a patient. We found a contract manufacturer. We worked with them to produce and validate these sensors. So a little bit of a non-traditional PhD and postdoc, but you'll find out in a couple slides it served me personally pretty well from a learning experience. So then the next step was, okay, let's actually start getting these into patients. So what were we setting out to do? Our goal was really a feasibility study. Could we get these into patients? Could they be measured within the workflow? Would we be seeing them on MRI? All of these things that we had done in animals before in Michael's lab. But now the question was, working within the clinical workflow of humans, how do you actually make this work? So the image you're seeing there on the left is actually, we were implanting 1 to 2 sensors in patients. And those 2 bright circles you're seeing, or 2 bright dots you're seeing in those red circles, are actually 2 of our sensors implanted into a patient that has cervical cancer. So we were able to meet the primary endpoint in 7 of 7 patients. We were doing this in the early days of the COVID pandemic, and unfortunately, our study was not deemed critical, so we shut down at 7 while our goal was 10. But in all 7 patients, we met the primary endpoint of feasibility. And for us, this was really the prerequisite to say, okay, what's the next logical step in what we're building here? And for us, that took the form of a spin out. So I'll now kind of talk about how the Bridge project has impacted how we think about this from a commercial perspective. Because I think one of the things we all really believe is that the research we're doing and the development we're doing should be translating into patient care, and startup companies are one of multiple vehicles to achieve that. So we formed Stratogen, and I think you'll start to see here up till this point I had spent till the Bridge project, I was kind of exclusively an MIT person, but this Bridge project really opened the doors to kind of get more into the Harvard ecosystem. The space on the right is an incubator space at Harvard where Stratogen is based, and it was the Bridge project that opened those doors for us to go there. So we started to take a step back and said, okay, we're taking this from research and turning it into an actual commercial product. What actually needs to be done to achieve that? And I've talked about the sensor going into the patient, and I've glossed over a little bit how you actually make the measurement. What we realized in this clinical trial was the measurement we need to make is actually not really a turnkey measurement off the shelf today. We realized we needed to innovate in the space of how we actually make the measurement to measure this sensor. And from our perspective, that started to open up a whole new learning opportunity. Something I really like to talk about how we learned from our own necessity that this had to be something that needed to be created. So I kind of will draw the distinction between 2 forms of MRI and why we actually need to do this. Because you might be wondering, MRI is everywhere. Why can't you just use the MRI as it is today to make your measurements? So 30 million MRI scans in the US every year, 95 million worldwide. And the issue is that the overwhelming majority of these are limited to qualitative scans, meaning the analysis is only visual or morphological interpretation. This can be pretty variable, clinician dependent, and in many cases lead to an incomplete understanding of disease or and that can lead to worse outcomes for patients. So the analog to qualitative MRI and the form of MRI we use to measure our sensors is quantitative MRI. This can give you a direct assessment of tissue composition. It can be used to make direct objective measurements of things like our oxygen sensors. But the problem is it isn't a scalable, harmonized solution. We can make it work at the Brigham, we can make it work at a couple other sites. But when you think about how do we give somebody a product that they're going to use, insert into a patient, not have our team in the room with them, not have our team at the scanner, how do you actually make that a solution? So, quantitative MRI has been around for a while, but the big issue is it hasn't been a practical solution. So we really started to innovate in this space now because we felt this was a prerequisite to everything else we were doing. Today, our solution takes standard MRI hardware and software, we collect images, and we're able to convert those into quantitative MRI scans. So we do this in a way that's much more harmonized and workflow compatible. We then can extract the biological information, and that could either be our oxygen measurements or many of the other measurements that you can use quantitative MRI for. We then produce reports or augmented images and feed that back to the clinician. We've really, from the start of this project in the Brigham, in the collaboration days here at MIT and over at Dana-Farber, we've really been hyper-focused on workflow compatibility. So we continue to do that because the barriers to deployment are much less when you're mindful about how does this actually get into the workflow. So we've been building this out, and as a company, we started to think, okay, should oxygen be the only thing we do with this? And I mentioned that quantitative MRI has a wide range of applications. So, we've started to think about what are other places that quantitative MRI, if we build this tool, should it only be a tool we use for oxygen sensing, or should we really think about how do we use this tool to impact patient care across a much broader spectrum? So we started to do research, and I think one of the things that was the core of this project back in the early days was going and actually talking to clinicians. So we continued to do that. We've talked to hundreds of clinicians over this project to really understand what are the applications of what started here at MIT and Dana-Farber and now has expanded into what we're doing at Strategen. And you'll see up in the top left corner there, solid tumor applications with tumor oxygen sensing. But you can see a lot of other applications that this technology can be applied to. And as a company, we've actually landed on one to start with. We're still working on the oxygen work in parallel. And I'll mention how we're going about that. But there's some applications here that are completely non-invasive that can get into clinical use in a very fast time frame. And the one we've actually focused on now as our primary go-to-market, putting on my, the terminology I use now versus when I was a grad student and postdoc here has definitely changed. I think much more in the context of, how do we actually make this sustainable long-term, is actually in prostate cancer. So right now, MRI is a mainstay in prostate cancer. And it is used to decide who needs a biopsy, and if so, where to target the needle. The problem is that it gets it wrong a lot of the time. 66% of the time, they're biopsying patients when they don't actually need the biopsy. And this leads to a lot of downstream consequences. There's pain, nerve damage. There's a risk of infection and sepsis and hospitalization. And we really started to see that, okay, again, MRI is part of the workflow. That's great. And MRI isn't getting the job done completely. So, avoiding those unnecessary biopsies, there's a potential to shorten the MRI scan protocol, saving patients and hospitals time. You can save money for the system here by avoiding these unnecessary procedures. And there's also a decent rate of misdiagnosis where the biopsy doesn't actually represent what's going on in the tumor when you look at the whole tumor. So these are all things that this platform we're building out can address. As far as where we are, I think it's always nice to kind of look back to, where is this going when we actually be using this in patients? We have a clinical trial coming up at the Brigham. So this involves collaborators that we formed as part of our Bridge project that we continue to work with today. And then focusing on getting this through the FDA clearance process and actually launching this commercially by late 2026, early 2027, following this up with all the other applications that I mentioned on the previous slide. So I'll close here with, I think it's always important to think about what is the impact of this project. And I think the products that we're producing are one way to think about that. We've since then gotten 2 SBIR grants from the National Cancer Institutes. Those are small business grants to continue innovation, research, and development. So Strategen has received 2 of those. Rob is a Brigham PI, or Dana-Farber Brigham PI on both of those grants. So again, this collaboration continues. There was another person on the Bridge project that left to go to Johns Hopkins right after we started. He's a collaborator of Strategen today. We have an advisory board member who we met through our work at Brigham. Brigham is the lead site for our first clinical trial and is really, Brigham Dana-Farber is really our home base. And then as we think about, what are some of the external recognitions? I think, I mentioned the impact this has had on me when I was a trainee. It led to opportunities for fellowships both here at MIT and at Harvard, focused on translating the technology. And then again, going back to this being the bridge into the Harvard ecosystem, we were named a grand prize winner of the Harvard President's Innovation Challenge for the work we're doing last Wednesday. So I think, really it bridged, I think the goal here was to bring together scientists and engineers from Dana-Farber and Brigham, but it really for us bridged us into this whole ecosystem of Harvard. And now we get to pull from where MIT is the best place, we get to pull from that. Where Harvard is doing something great, we get to pull from that. So I'll close here with just saying that, I think from the perspective of a trainee, this was truly a transformational opportunity for me. I think this really was a springboard into, what I've been doing now for 7+ years. It brought us great people to work with, and really truly grateful for the funding opportunity and excited for the work we've done and the work we're continuing to do, and just seeing the acceleration of what we're able to do here and what we're going to achieve over the coming years. So with that, I'll close, and I'm happy to take any questions.
Greg, that was terrific. Thank you very much. I'm actually going to start the questions by asking for comments, actually, from Robert and from Michael. Just from your perspective, why don't you start? How did it go from your perspective?
So I think this was a great opportunity for us to bring in some technology that wasn't at the time available at Dana-Farber. And so this was something that both Larissa and I looked forward to because it was addressing a very well-known but not measurable clinical problem that was something that's a challenge in radiation oncology.
Well, of course, I've been lucky enough to work with a great group on this project. Greg in particular has just done a stellar job. He didn't really explain making these catheters involved going to a contract manufacturing organization and basically renting their quality system. It was Greg that was there every day for months and months to produce the catheters we needed for testing, sterilization validation, all of those things went through just so we could use these in patients. And I think it was interesting to watch the experience of our clinical collaborators too. They got introduced to what it's like to develop technology. I remember Larissa coming over, going to the 6th floor, and we were concerned about modifying these catheters and making them weak so that they couldn't be inserted into the patient. And we didn't know what forces it would have to undergo. So she would come here and we would take a piece of steak on a load cell and she would push them in. And she'd think, what am I doing here?
I guess to build on, I remember showing up at her office with pork, chicken, and steak and said, which one of these feels more like a cervical tumor? And we picked steak, but.
Though following up on that, Larissa was the only person in the world ever certified to use this technology.
Yeah, there was a training part of it. So she was trained to use these specific catheters.
Questions from the audience? I'm not seeing hands raised. Now I am.
That was a really interesting talk. So I was interested in whether, considering tumors are highly hypoxic and that hypoxia can affect how tumors grow and progress, I was wondering, have you considered or applying your sensor to predict whether tumors would respond to certain drugs or chemotherapies that are anti-metabolites and that would affect their proliferation?
Yeah, definitely. So, we focused on the radiation oncology application for a couple reasons. One, low oxygen tumors are less sensitive to radiation. You can boost dose to try to overcome that resistance. But it's not the only place in oncology where it plays a role in chemotherapy, immunotherapy, how well oxygenated a tumor is definitely plays a role. So we viewed this as a kind of tip of the spear but by no means the last application within oncology.
Yeah, I'll just add, there are plenty of programs that have failed with drug therapies that are targeting hypoxia, primarily because they don't know how to stratify patients, who's truly hypoxic and who's not. So this could be a means by which to stratify those patients and show whether they're
It's great to see how this has progressed. Thanks so much for bringing back the story. Did I understand correctly that you are able to do analysis on MRI data that's coming from existing MRI machines? So you're basically, this is open to the world who's doing MRI.
Correct. With about 10 to 20 minutes, we can have a scanner configured to do what we needed to do. And in many cases, the acquisition or portion of the acquisition is actually part of the existing clinical protocols. So it's very workflow compatible from that perspective. No new hardware, no new software required. So pretty quickly we could deploy and have anyone anywhere using this.
That's incredible. It's
one
impactful.
of the difficulties, though, that the company is addressing is that comparing scanner results from one scanner to another comparing one scanner to another is difficult. It's not currently done. I mean, that's why this is a research area. What the company's doing is showing how to translate results from one scanner to the next, or from one scanner on day 1 to the same scanner on day 30.
Wonderful. Let's thank this team one more time. So sort of transitioning from what you've just heard to what you're about to hear, I think these two talks were examples of sort of an amazing application of sort of cutting edge technology to a clinical problem for the first time. It's like, I can't believe you can even do that. You'd even think about doing that. But here we see examples where it's possible and actually practical and now in patients. The next talk will represent innovation, an old idea that you can stimulate the immune system to recognize and eliminate cancer, but a need to do it better. And here we'll have a team represented by Kathy Wu from the Dana-Farber and Brad Pentalute from the chemistry department here at MIT and the Koch Institute. I'm not sure the order, but I'll let you folks take it away.
So great, thanks for the introduction. It's such a pleasure to be here and incredibly grateful for the support and what it's provided for us. So I will say that some of the challenge has already been discussed before because we're talking about cancer, but just to restate it, I think that the foremost challenge that we face as investigators, clinicians, is that cancer has heterogeneity. This is the fuel that drives resistance and relapse, and this has really been brought home to us by all the genomic sequencing that we've done. Cancer doesn't exist in isolation. It coexists with the host immune response. But unfortunately, by the time that we diagnose our patients, often there's all sorts of mechanisms that have been put into play to inactivate host immunity. This type of conundrum was recognized very early on. So even back in the 70s, Noel, at the first very moment that we understood that cancer was fundamentally a disease of mutations, somatic mutations, there was this understanding that this was going to provide a therapeutic conundrum for us. And he stated at that time in the 70s that immunotherapy becomes a leading candidate for the easiest means of destroying the remainder of the neoplastic clone. So fast forward to where we are right now, we've recognized that at the center of this kind of cancer immunotherapy revolution that we've been going through, that T cells are really center stage in this work. T cells have many different capabilities and witness all the different types of therapies that are out there right now, each trying to harness a specific aspect and functionality of T cells. But what I'm going to talk to you today is about vaccines, because vaccines do, amongst the different modalities in the magenta box, it has the ability to stimulate either pre-existing T cells that are in the tumor bed, so those are the pink cells, or stimulate de novo anti-tumor T cells. So basically building up the army of T cells that can go at the tumor. So reinforcing what you have, but bringing in new ranks as well. And so as Tyler mentioned, vaccines have been around a long time. The concept of cancer vaccines have been around a long time. It's really been a roller coaster in terms of how we think that this field has evolved. But I think that what I hope I'll convince you today is that we've made quite a bit of progress. We're here at the Bridge Symposium. This is our team member Ed Fritsch really loves the Golden Gate Bridge. So this was a figure from a review that we wrote with the red pillars. But we tried to think about what are the key things. The thing about vaccines is it's actually a pretty complex therapy. There are many different components. And what our Bridge project has been really focused on is thinking about the antigen. So what do we have to target? And also, how do we deliver? So how do we get the vaccines in there? And this was really the nidus for thinking about how to bring our project together. I'm speaking on behalf of Patrick Ott, who's an extraordinary melanoma clinical investigator who couldn't join us here today. And I'm here with my partner, Brad. So we'll get started. So bringing this team together to address the challenge, I think that once we had this funding, we actually had for quite some time meetings, kind of a club that we would meet over at the Broad, people from both sides of the river coming together and really trying to discuss all these different complex aspects. And members of those teams still in different iterations continue to meet today. I want to say that a focus of our work has been neoantigens as immune targets, and this is also an older concept, but a convergence of transformative technologies has really made these targets a reality. The first is that sequencing has become available. The second is improved epitope prediction. And the third aspect is the availability of immune checkpoint blockade, which is really the founding by Nobel laureates and other esteemed members of our community. And I like to liken this to that Obi-Wan Kenobi moment where the Raider shield goes down and allows us to really kind of start to target the Death Star or cancer. And so we very early on, recognizing that next generation sequencing was available to us in our community, we began to realize that those mutations that we could discover by sequencing, at least some of them would generate mutated peptides that could be processed, presented on the cancer cell where they could be recognized on the surface by T cells. So these are neoantigens, and we thought that this would really be hitting the sweet spot. So not only would these be exquisitely tumor specific in expression by virtue of being born of somatic mutations, but also because they were somatic events, that they would be highly immunogenic because of a lack of central tolerance. And this is akin to pathogens and really different than where we had been decades before, which was thinking about targeting shared overexpressed tumor self-antigens that were variably tumor-specific, variably immunogenic. And really, we're at a day and age where we can actually take the data, string together our computational pipeline, so it's actually very feasible to go from sequencing data, find those mutations, perform those predictions, and on the back end, come up with a list of targets that are predicted to be immunogenic on an individual basis. So just to reiterate that the challenge up until now was how could we find those mutations on an individual basis, recognizing that every patient's tumor is different, going back to that theme of heterogeneity. And so I think this was a paradigm shift. I think this idea that we could launch a multi-pronged attack against a person's own neoantigens and spare native tissue and really try to get in there. And so what the Bridge Project enabled us to do was to bring different teams from both sides of the river, all the kind of technological and device sort of expertise on one side, and really our clinical infrastructure on the Dana-Farber. And so just to, again, briefly reiterate, leading up to this time, we were already able to kind of conceptualize this idea that we wanted to go from individual patient to mutation identification, to thinking about how we could synthesize a panoply of mutated peptides that we could get into a syringe and give to patients, hopefully together with immune checkpoint blockade. And I'm not going to go over this in depth, but just only to say that we've been able to perform proof of concept studies using our clinical infrastructure to demonstrate that this is highly safe, feasible, highly immunogenic, and promising in combination with immune checkpoint blockade. So the Bridge project has been enormously impactful, and I appreciate Jeff's previous slide because the impact on trainees is tremendous. This has led to junior trainees becoming faculty members or joining in industry in really important positions. It has led to numerous federally funded and foundation grant applications. But I want to give you a sense of kind of the directions that we've taken from the Bridge project. So the first, because this is a genomics impacted sort of therapy, it was really important for us to get antigen prediction correct. And so the Bridge project directly helped to fund, or we were among the first to use cell lines with single allele expression to generate large swaths of data from mass spec that we could use to train our models for prediction. And this has led to new models, we call it HLA Athena, that have been favorable in comparison to other existing models. And this is the prediction program that we are using in our current clinical trials. It has also directly fed our infrastructure for our bench to bedside and back again efforts, and in particular, disease-specific research, so we could understand better what is the hostile environment that we're sending our vaccines into, and also to capitalize on the biospecimens that we're collecting from our patients so that we can do the best we can in terms of human immune monitoring. And just as an example, we've really been focused on trying to, at a single cell level, understand the phenotype of T cells, link that to the kind of clonal architecture of T cells based on that we can detect based on their T cell receptor, but importantly, what are those T cells seeing? What are the antigen that they're going after? And how do we make all those linkages together? Another aspect is how do we actually visualize those T cells within tissue? So are those T cells that we want, that are neoantigen specific, are they actually getting to the site of tumor? Are they actually collaborating and cooperating with other members of the immune system that are in the environment, and what crowd do these T cells run in? And then I'm going to pass it to Brad because part and parcel of our effort has been really thinking about improving antigen delivery, and I mentioned that our direction was to think about synthesis of mutated peptides.
Yeah, it's great to be here, and just to add a little commentary to how I ended up collaborating with Kathy is she heard that I like peptides. And so I think she was across the river in an office one day and said, "Hey, Brad, can I come over? I want a bunch of peptides. Other people can't make these peptides. I really want to understand how I can use them as a vaccine in personalized cancer therapies." And I said, "Tremendous, come over." And so all of a sudden we're in my office and I'm showing them the robots and everything that we can do. And she says, "You know, we can really do something here. We can get these materials that are really hard to get because they're so-called long peptides, which turned out to be difficult for CROs and stuff to produce. And if we could perhaps maybe even get them in a day, we could start to understand what's going on with these personalized cancer vaccines." And so that was the genesis of our project. And we all came together and we said, okay, let's figure this out. And my role was, the first stage was to figure out how can we actually manufacture these long peptides in a matter of minutes. And moreover, if we started to look at these personalized vaccines that needed to be generated on a patient-by-patient basis, and because the prediction algorithms needed to be improved, we needed to make 30 or 40 of them. And then she started talking to me about, can we actually do this in a day? Can I give you the data that we learned from a patient, and you can go print them off in a day, and then we can go back and do these vaccine studies and all the other science that's necessary to really unlock this paradigm. And so right away, we went ahead and said, okay, well, could you have peptides that you've already injected in humans? Can we see if MIT and their robots can actually start to do better in terms of having less likely of failure, breakdown in the chemistry, and actually also be able to do this really fast? And so you'll see the machine over here that we have at MIT in the labs. We have about 4 of these. There's a video if you can go ahead and play that. And what's really, really great and what accelerated the pace in which we did our research together was we went ahead and printed off all these peptides that were predicted to be viable vaccine candidates. And we did it with a very high success rate, a success rate that others couldn't actually achieve. And what's neat here is that this instrument that we have can make these long peptide vaccines literally in 20 minutes. in terms of synthesis time, and then there's downstream processing that requires some additional effort. But at this time, we're not actually, unfortunately, typing in a vaccine. We're typing in GLP-1, this remarkable weight loss drug. But we can make that in 15 minutes. This instrument's fully charged with amino acids. So this actual reactor will give you enough material to go all the way to the clinical trial. So you make multi-milligrams, and so more than enough for a vaccine study. And so the machine actually just marches along. It strings together amino acid by amino acid, eventually arriving at this antigen. And so you actually also get a ticker tape of synthesis. It tells you, okay, that amino acid was delivered, and it also reports on some of the chemistry, the efficiency. So it turned out to be quite powerful. And this was the first publication that we actually put together, and a lot of learning enabled us to dive deep in some various vaccine areas. And so as Kathy mentioned, we really wanted to focus on bulletproof manufacturing, but then also start asking questions, can we actually use engineering to get the antigens to the right cells? And how does that affect the vaccination? And in this study, we actually said, well, we want to get those vaccines directly to dendritic cells because we thought that would be most important. And moreover, we want to get those antigens directly in the cytoplasm and change how they get presented, presumably by an MHC-1 process. And so we built a nano machine based on anthrax toxin. It sounds a little bit crazy, but we rendered it non-toxic, and we used it to target a specific receptor, as far as we can tell, that's unique to dendritic cells, and we had a targeted delivery mechanism to get these antigens into those dendritic cells, and then followed up by doing actual animal models, and a myeloma model, as I'm showing on the right panel. And lo and behold, we were actually able to slow down tumor growth by virtue of direct delivery to dendritic cells, these antigens that actually would stimulate the immune system. So we also adopted another approach to understand delivery. And this actually was even a broader collaboration, which I really loved about the Bridge project is literally you can go knock on anyone's door and say, we have this wonderful mechanism, all these great people, but we also need your help. And so this was an expansion of the Bridge project with Darryl Irvine in addition to our team. And this question was, okay, we're learning that we can do targeted delivery of vaccines, as I just showed you, to dendritic cells. But can we actually even have a simpler delivery system where we start to take what's called cell-penetrating peptides and use that to really boost presentation and ultimate immune response? And what I'm showing here is that if we attach these cell-penetrating peptides to known antigens and actually inject them in animals, you actually get a significant boost in T-cell response. And so these experiments are still being investigated. And we think it's because we actually get the antigen into the cell, into the cytoplasm, and change presentation by virtue of promoting an MHC-1 presentation. And so this is another delivery effort that we went after with the Bridge effort. And so the last part that was actually really unexpected. We started asking the question, now we can get antigens into the cytoplasm, and the way they get degraded and processed is all by the proteasomal machine. And we asked the question, how does the actual sequence of the antigen affect whether or not it gets presented? And we started to see a real strong pattern that no one really had identified, which is the C-terminus of the antigen plays a strong role of how the proteasome processes it and then eventually presents it and stimulates the immune response. And it's really complex, right? Where there's 20 amino acids, there's so many possibilities of how they can be actually strung together on these antigens. And so we had to build a machine learning model to actually allow us to predict whether or not particular sequences were likely to be processed and how that correlated with whether or not they were presented and stimulated the immune response. And we actually built a really nice machine learning model that actually starts to allow us to understand how the C-terminal region of these antigens actually promote presentation. And in particular, if you have high likelihood of degradation in the cytoplasm, actually we found you get a much stronger immune response, as I'm showing over here for a model, OVA antigen, but then also over here a GP100 myeloma antigen. And so it's quite exciting. So we're starting to bring in cutting edge machine learning into this overall space combined with some of the technologies that we developed. So I'll turn it back over to Kathy.
So I'm cognizant of time, so I'll just share some final clinical avenues that we've been undertaking. I just wanted to add that the other aspect is that we've continued on trying to iterate in terms of our clinical trials beyond our first set of 2 studies. And this has really been a pleasure to bring in new technologies, new ideas in each disease that we start to think about. We think about new opportunities and really trying to do the best we can in terms of our learnings. And I just want to share with you some recent results that we're excited about. This was work by David Braun, who was a trainee and now is an assistant professor at Yale New Haven. And he really spearheaded a trial in patients with advanced renal cell carcinoma, following on using synthetic long peptides that were given to patients in a prime boost sort of schedule. And the results have been really good. These patients did super well. A small study, but really going out 3-4 years, a paucity of recurrences, and otherwise what are patients with really advanced disease, no toxicities. There was unfortunately one death related to mental health rather than treatment or RCC. Robust immune responses, including against what we think about drivers. And so that's been exciting. And then if you follow the red line, we see the persistence of vaccine immune responses over years. And then finally, we detect responses against the patient's own tumor cells, which of course is what we want. So I think in the final sort of slides, I just kind of want to give you a perspective of where we're going as a field for personalized cancer vaccines, a field that we really were at the inception of. And it's really, whether with peptides, but also these days, we also think about mRNA. Across the various studies that have been going on, we do see promising clinical results with decreased recurrences. We've been able to track T cells that are specific for neoantigens to the site of tumor. There has been evidence of epitope spreading, lack of recurrences after adjuvant vaccines, even in really difficult to treat cancers like pancreatic cancer. And what's been very exciting is in this past year, it was reported out a randomized open label phase 2 trial, so in melanoma. So a first time ever where we actually see benefit of the vaccine together with a checkpoint blockade in comparison to immune checkpoint blockade alone. So we're really now, the field is really eagerly waiting, fingers crossed, in terms of studies where we have hundreds to even thousands of participants that are enrolling or starting soon. So I think this will really give us a handle on where we're going with vaccines. I've had a lot of great trainees, but I've also been able to engage in even young kids who seem to be interested in the field of cancer immunotherapy. So this was an 8-year-old daughter of a colleague who said in the end, I think you should also get it through a shot. So that was her advice. And we continue to think about how we can impact our trainees. We think about how we can give vaccines. Really, it can be given across different moments in cancer, whether it's advanced disease, prevention of relapse, or cancer prevention. And I think there's unique challenges in each of those areas. But I do think that in the not too distant future, I hope our patients can just say, please, can we just order up my cancer vaccine that's personal to me and come back in a couple of weeks and it'll be ready in the clinic. So thank you for your support. It's been great.
Thanks, Brad. Thanks, Kathy. We have time for questions from the audience.
I like the last slide.
Looking out over sea of faces. I'll start with a question. Many of folks in my lab are thinking about not just CD8 antigens, but CD4 antigens. You use long peptides. They could elicit CD4 responses. Can you just comment on what do you actually see when you put these things in?
Yeah, so we do see, for at least not just in our long peptides, but also in the mRNA experience, it's really been CD4 responses that seem to dominate. A lot of those by transcriptional sequencing do seem to have a cytotoxic profile, which sounds promising. We have not explicitly tried to elicit a CD4 response because we're getting them as part of the ride anyway when we're targeting CD8s. I do think there's a lot of literature to support the need to have both humoral, both CD4s and CD8s to really generate a robust immune response. One of the questions I have, and certainly the mouse models are a great place to look, is what's happening in the periphery. A lot of our monitoring is in the peripheral blood, but what's happening at the site of tumor. And I think that perhaps we want our CD8 cells to not be hanging around in the blood, but actually at the site of tumor. So I think that's something to look at.
Great. Any other questions? Okay, seeing none, thank you both. And we'll bring this part of the session to a close with our fourth presentation. I'll just mention that we're featuring 4 long talks. We could have featured 40 long talks from the great work that's been supported by the Bridge Project over the years. We actually chose to feature high technology talks, and that was in part because Art himself was very into technology. He was a technologist himself. He thought the T in technology here at MIT made a big difference in what we could contribute. We contribute a lot of science as well as technology, but we featured technology today in part for that reason. And among the technologies that Art was most excited about was computational technologies. That the power of computation could change the way we think about cancer and manage cancer. And our final long talk will be in that area. I'm not sure, I see Regina sitting over there, which makes me think she's maybe not speaking. But the team will be represented by Regina Barsley, our colleague here at the Koch Institute, and Alicia Sequist from MGH. And they're going to talk about applications of artificial intelligence on lung cancer screening. This group came together first, but with Footbridge funding, actually. And based on the success of their Footbridge work, they've expanded and actually today still have a traditional grant in this space. So Alicia, take it away.
No. Try again now? Okay, great. All right, thank you. It's a real pleasure to be here on behalf of myself and Dr. Barsley to tell you about what we've been doing now for the last maybe 5 years, trying to come up with better tools for lung cancer screening. So I'm a medical oncologist. I work over at Mass General. I take care of patients with lung cancer all day. And many people do not realize that lung cancer is actually the leading cause of cancer death for both men and women, not just in the US, but worldwide. This is often not spoken about in public as much as some other types of cancer. But one of the groups with the fastest rising rate of lung cancer in the US is actually younger women who have never smoked. So I think this is really a problem that we, all need to think about. For about 10 years, we've had a screening test available for lung cancer screening, which is a CAT scan of the chest. And when we look at the low-dose CT scan, as it's called, for lung cancer screening, and we put it against some of the other cancer screenings that we have, it actually performs very well. The number needed to screen, or NNS here, is a number of people that you need to screen with this screening test in order to save one life from cancer. And so you can actually see it's quite a bit more efficient in terms of a screening test than mammography, colonoscopy, or pap smears. But unfortunately, it's in the implementation of this test that we have failed. So this is now a few years old from 2020, but this is basically a bar graph of all the different states in the US and how many of the people who are eligible for lung cancer screening are actually being screened. And so it may be hard to see the initials of all the states, but the US average is 5%. We're screening 5% of people who are eligible. Massachusetts is actually in the lead. But you have to look over at the y-axis to see we're only doing 15%. So even though our bar is really high, it's actually pretty low. So the implementation of this, the rollout of this screening test over the last decade has been really poor. And especially when you overlay where lung cancer tends to occur the most in our country, the darkest blue areas that have the highest deaths per 100,000 from lung cancer also happen to be the areas where lung cancer screening is the least available and the least accessible. So there's a lot of problems with the way that we're currently doing lung cancer screening. Looking at this problem from a slightly different perspective, the one that I see in the cancer clinic taking care of lung cancer patients, instead of thinking about all the people that could have screening and how we're doing screening them, let's look at it from the perspective of all the people who are actually diagnosed with cancer, and would we have been able to help them if we had better implemented this screening test? So on the left, the highest bar represents the number of patients who are diagnosed in the US with lung cancer each year. And then the second bar, which you can see is less than half the height, is those who would have been eligible for screening under our current criteria. So our current criteria, you have to be age 50 to start screening for lung cancer. We know many people are diagnosed younger than age 50 now. Also, you have to have a specific minimum level of tobacco exposure. And we know that more and more lung cancer patients are actually not meeting that criteria. Either they have never smoked or they smoked in college a long time ago, just a little bit, and it's been decades since they smoked. They don't qualify for lung cancer screening the way we have it set up. So more than half of our patients who actually have the disease are not even able to access screening if they had wanted to. And then, of course, the second big drop-off shows among those who are eligible, how many are actually participating. So this is a big failure, just across the board. And so the way I think about at least two urgent needs are we need to improve our assessment of who's even at risk for this disease. We have these screening criteria, but when you look at the people who actually have the disease, more than half of them wouldn't have qualified as high risk enough to get the screening, when actually we know they're 100% at risk. They got the diagnosis. Then we also need tools to help us get individuals to screening and stay engaged with annual screening. All of this is important because lung cancers are often without symptoms until they are quite advanced. And so if we want to be able to try and diagnose patients at a time in their disease course when they're still curable, it's very important to get to them before symptoms develop. And the only way to diagnose someone who's completely asymptomatic and feels well is to do a screening test, looking at a seemingly healthy person to see if there is a hidden cancer. So in the past, there's been a lot of research done with risk models, or really sort of clinical calculators, these types of formulas that you can plug in, things like the age, gender, the race, and different clinical factors, and it will spit out a risk score of whether or not this person, based on epidemiologic data, is at risk for cancer. And for lung cancer, this Tamamaji model, or also called the PLCO model, is probably the widest used and validated worldwide. But it was really developed based on epidemiologic data from the end of the last century, when it was much more clear that tobacco cigarette use was the primary cause of cancer. And so you can see that many of the 11 factors that get plugged into this calculator have to do with cigarette smoking. And we know now that the epidemiology of lung cancer is changing. And I show the cover of an article of an issue of the journal Nature from about a year ago that had a very, very impactful article from the UK, from Charles Swanton's group, showing how air pollution is actually driving, or maybe driving, a lot of the lung cancer globally. That is completely independent of tobacco use. And it gets complicated when you're trying to think about how would you make a clinical calculator that someone would put in their exposure to air pollution. I mean, we could talk for an hour about how inaccurate it is when people try to recall their cigarette smoking history, but at least that is an active act that they participated in. It's impossible for any of us to know really what our exposures to different air pollutants has been throughout the course of our life. So, we need predictors that don't rely on an individual's recall of what their life exposure has been. And so I was very, very lucky and honored to meet Regina several years ago now. We met sort of twice in rapid succession. One was at a Stand Up to Cancer meeting where we were talking, about some of these problems. And then shortly after that, she had a friend who was a woman who had never smoked and was diagnosed with lung cancer, and came to see me for a second opinion. And so that kind of brought us back together, talking about, how this problem is really important and really widespread, and most people don't know about it in the public. So with that enthusiastic collaboration, we set out to use her background in developing AI models off of radiographic images, and we designed this model, which we named Sybil. So Sybil is is a AI algorithm, a machine learning algorithm that basically can look at a CAT scan. So a CAT scan is actually a series of images, kind of depicted up there on the left, that shows when you put them all together, you get a three-dimensional space of what the patient's thorax looks like. And Sybil looks at all the information that's in there, the anatomy of the lungs, the heart, the bones, the muscles, everything that is imaged in a chest CT. And through training it with over 44,000 scans from a large clinical trial where we knew what happened to the patients later, we knew which ones ultimately did get diagnosed with lung cancer and which ones didn't. The model was trained to be able to predict which patients were at high risk for lung cancer and which ones weren't. This has a lot of practical, the way that Sybil works has a lot of practical advantages. So there's no image annotation that's needed. You don't need a radiologist to circle an area of question. You don't need to have them spend time telling the model, please look at this and give me some more information. You don't need to plug in any clinical data. So you don't even need to put in their age or their sex. You certainly don't need to gather things like smoking history or any other type of exposures. You get an instant readout. It gives you multi-year future risk stratification. And I think one of the most important innovations is that this is an open source code. It's freely available for people to use and innovate on worldwide. So about a year ago, we published our initial results, and I'll summarize that here with these 3 graphs. So these are 3 different validation cohorts that we studied. So after the model was trained, we exposed it to 3 different populations of CAT scans where, again, we sort of knew the answer. We knew which patients would go on to develop lung cancer and when, and we knew which ones did not. And so panel A is more scans from the original study in which the model was trained. So these were not part of the training set, but they were from the same overall study. And each of the different colored bars represents a year of prognostic predictions. So Sybil spits out 6 numbers. What's the risk of cancer in 1 year, 2 years, 3 years from the time of this scan? And so each different color bar shows the curves of how well the model is distinguishing for those different years out. In the center in panel B, this is a cohort of patients from Mass General who were part of our regular screening program. So in general, panels A and panels B are somewhat similar types of people that are represented with those CAT scans. They're all US based patients, North American. They're predominantly white, and they are heavy smokers in general. They have a high tobacco exposure because that's how the trial was set up. And then that's also how our clinical programs have rolled out across the US currently. So this is very similar populations that show the model works well. What's really interesting though is panel C is very, very different. So panel C is a group of 12,000 scans that we got from Taiwan. When we think about air pollution and cancer in people who haven't smoked, this is an extremely, extremely prevalent problem in Asia, and especially in East Asia. And so some countries and health organizations in East Asia, and particularly the entirety of Taiwan, now have screening programs that are just for everybody. So any adult can go in and get a lung cancer screening. And so here we have really more of a population view. Some of these patients have tobacco exposure, many of them have none. And you can see that Sybil is performing very well in this Asian population. So that gives us a lot of hope. So the clinicians that hear about this work, they all ask the same question: what is Sybil seeing? Because radiology is really such a key part of clinical medicine, but it's all about what you see. It's all like the art of being able to differentiate. Is there a pneumonia there? Is there cancer? And this is the most common question. So. This is a machine learning algorithm. We don't know exactly all the factors that go into the model. We know that it doesn't approach it the way that a human radiologist does. And here's an example that often helps clinicians appreciate that this does something totally different than a radiologist. So this is one slice from the right lung of a participant in the clinical trial that we used to train the model. This is a 69-year-old male who has long history of smoking exposure, and this scan was read by the human radiologist as negative screen. There were some minor abnormalities, but nothing that was suspicious for lung cancer. But Sybil looked at this exact same baseline scan and placed the patient in the 75th risk percentile. Now, in trying to understand what is Sybil looking at, we've been working a little bit with something called an attention map. So we can ask the model, okay, you gave this risk, what part of the anatomy did you spend the most attention in calculating that risk? And so it can produce this sort of red-toned map, which is, for any of the clinicians, this is not a PET scan. It looks very much like a PET scan with that red hue. But the attention map for Sybil showed that a lot of the attention was paid by the model in this anatomic area. And when the radiologist goes back and really stares at that area, there's nothing that looks suspicious there. But when the patient came in for their next scan as part of the clinical trial, there was actually a mass that appeared in that exact same spot. And again, in retrospect, there was nothing there. Sometimes in retrospect, we can say, oh yeah, there's a tiny little dot, but not in this case. So in this case, the patient underwent surgery. This was a cancer. It was a stage 1 cancer, and hopefully this patient was cured. So this was, a win for your standard radiology screening and for the trial. But in the real world, what we know as lung cancer screening has rolled out is a vast majority of people, once they get a clean negative scan, they don't come back the next year. So this guy was participating in a clinical trial. The adherence was very, very high in that trial. He did come back. He was able to have an early stage lung cancer detected. But in the real world, chances are that we never would have seen him again until he had more advanced cancer. So can we use this type of a technology to personalize screening for lung cancer and take away, in that case, some of the barriers that have come up? All of this, we think that a lot of the reason why so few people get screened for lung cancer is that the questions you have to ask to figure out if they're eligible, you really have to grill them about their tobacco history, and I think it can be uncomfortable for both the clinician and the patient because of the stigma surrounding smoking. So a lot of doctors just don't ask, they don't bring it up, and no one goes for screening. So, can we use a tool like this to take away the stigmatizing questions about smoking and just make this something that everybody gets? And then the subsequent screening schedule could be based on their personalized risk, completely separate of whether they've smoked tobacco. This would put lung cancer screening in the same paradigm that we screen for other cancers. So colon cancer screening, everyone gets a colonoscopy at age 45. If there's no polyps, you don't have to come back for 10 years. If there are polyps, you may come back quicker. Same with cervical cancer. It's highly tied to HPV testing that goes along with the pap smear, and the interval of repeat testing is personalized based on your individual risk. So that's what we're hoping. Our next steps, this has been such a wonderful collaboration that Regina and I and our whole team have had. We've been lucky enough to have been featured on a lot of national television shows in the last year, so NBC Nightly News and Nova. We have launched a large multi-center Sybil implementation consortium with some colleagues in Illinois, Tennessee, and Georgia. And then also the VA, the Veterans Association, has a large project to integrate Sybil with other orthogonal biomarkers, which is being led by Steve Dubinette at UCLA. But my very favorite part about this whole project has been our team and the friendships we have made, both across the Boston community, between Mass General and MIT, and now nationally with some of our collaborators like Rhea Saragabon in Memphis and Mary Pasquinelli in Chicago, and we have wonderful trainees too, like Dr. Chang, who's here today. So thank you to the Bridge program and to everyone that has made this work possible. Thank you.
Thanks very much, Alicia. We will have questions, but before we do, I'm going to ask Regina, if you don't mind, to comment from your perspective how this program has impacted what you do from your perspective at MIT.
So I just decided that Lisha does such an amazing job speaking that I would never match up as a boring computer science professor. But at any rate, Lisha and I had this vision of taking the idea of doing prevention and being able to assess the risk. I've never worked on lung cancer prior to meeting Lisha. It was really based on the friend's experience. And we didn't have any chance to apply for outside funding. Lisha didn't do AI, and I didn't do anything for lung cancer. We didn't even know if it is even feasible and it will work. Because in contrast to mammogram, which is accepted way of doing screening, the whole population, we didn't know if it has a chance to work looking on CT scans. So Bridge gave us a lot of hope and funding to start our project, to bring our trainees. And within very short time, we see that it's promising. We brought other people on board, and we are continuing this collaboration. I just want to add to what Lisha said, that in addition to collaborators within the US, there are multiple countries that are currently exploring how to adapt CBEL for national screening. One example is Croatia, another example is Greece, Saudi Arabia, where everybody, the same way as in the US, are dealing with the question who should be screened and how. So I'm really humbled that I had an opportunity to collaborate with Lisha and her team. And Bridge really gave us an opportunity to translate the science into, concrete clinical outcomes and hopefully change, the way lung cancer will be diagnosed in the future.
Thank you very much. I did see a couple of questions here.
Thanks, that was amazing. Two quick questions. One, does the 1 in 300 screen, does that include the recent data on perioperative checkpoint blockade for, no?
No, so that's a good point, which is that therapy for early stage lung cancer has been improving in leaps and bounds. So it's even more urgent that we find it at the early stage now because we have a higher chance of curing it today than we did even 3 years ago.
Yeah, so that then leads to the next question. I don't want to sound like a Merck shill, but is there a population of patients? So for example, for DCIS of the breast, patients are given tamoxifen, even though only a very small fraction of those people will ever progress to a lethal breast cancer. Is there a population of patients who you would consider a randomized trial for PD-1 blockade, even before they have a identified lung cancer?
I think that using CBEL to find out who's truly at high risk could really help as an eligibility criteria for such a trial, because using tobacco exposure as a risk to do such an interventional trial on, I think is just not good enough. There are people who have a heavy tobacco history and never get lung cancer, and there are many, many people who won't meet your criteria that will get lung cancer. So yeah, we think that's actually a great use of CBEL.
Yeah, great talk. I'm curious how you think this could roll out, because trying to read your receiver curves, it's like 90% specificity was probably where you're at. And if you try and roll this out to just about anybody to be screened, that's it's
a big leap.
You're going to have a very high false positive rate, right? Even 90% specificity. So the predictive value, positive predictive value would be low. And so then what do you do? I mean, because a positive doesn't really mean that you're likely to be positive.
Yeah, so these are great questions. We've found that rolling it out is not as simple as it may seem on first pass. Even just I'm sure your work is similar, even just the downloading of the tool into a hospital environment has a lot of challenges and a lot of security concerns from the hospital environment. So, that's why we think that some of the work we're doing in our implementation consortium is actually so key, because we're literally going through all the steps and trying to put together a how-to guide of things as simple as downloading the tool, but then the much more complex questions of, well, what do you do with the results? One important thing is figuring out a threshold. Regina wants to talk too. Figuring out a threshold of when do we act. So I think that false positive rate will be very dependent on kind of the threshold that you set for what is a high risk score. Do you want to say something?
Yeah, so I'm very passionate about this question, because every conversation with clinicians after they decided how to install it and whether it works is what is a threshold. And it seems to me very analogous to discussions that's going on in the breast cancer community. When do you start screening? Do you screen every year? Do you screen every 3 years? There is a whole population of mammogram deniers that often publish in New England Journal of Medicine that have a lot of thoughts about, over medicating the population. I think that the tools like Sybil, and hopefully we can incorporate other markers within Sybil to make it even more precise, I'm talking about blood biomarkers and other things, they would enable us to discuss the strategies much more scientific terms, not just a policy and opinions, and you can decide what is the right threshold given, the budget in the population. But unless these tools become very precise and they work consistently across different populations, it's just going to be a matter of debate and not a matter of science. So we are currently trying to see how different groups are thinking about it. There is a lot of varieties, but I think that once we consider different alternatives, see what exactly it produces in the population, then, each group can decide for itself what is the right way to continue. But definitely not stopping it because we cannot agree ahead of time what is the right strategy.
Yeah, thanks. Alicia, that was absolutely beautiful. Here. I'm just wondering, is there something peculiar about the anatomy of the lung in terms of it being air-filled and just the level of resolution of these CT scans that you think that this is something that would be unique to lung cancer screening versus, for example, devastating cancers like pancreatic cancer, ovarian cancer, where we just really, again, similar problem where there's, patients are often widely metastatic before they're even diagnosed. Do you think that this kind of approach could be used more broadly in cancer?
I think it could. Regina's group has done work with mammograms. She's briefly referenced, but she developed a model called Mirai that uses two-dimensional imaging of the breast to do very similar work, and that has been validated in hundreds of thousands of patients worldwide. And I don't know, do you want to talk about the prostate stuff?
Yeah, so I just want to know that when we compare the accuracy between mammograms and CT scans, we see, even though the two different organs, that on CT scans for lung you actually get much higher accuracy because it is higher resolution modality than mammograms. We have initial results on prostate. The previous speaker was referring about unnecessary biopsies. We can demonstrate from MRI of prostate, you can actually decrease unnecessary biopsies. We're starting another work with MGH on pancreatic cancer, where there are a lot of MRIs to predict the future. So I think that if you can image the organ with high resolution, we can make these predictions. But some organs you cannot always image so well.
Okay, we'll stop there. Let's thank our speakers one more time. I will point out that, as I mentioned, Art in particular is excited about things that we do here on the computational side. Each year we name one team for Art and Linda. The Gelb team, Leisha and Regina and their colleagues were the Gelb team a few years back. So I wanted to make that point. We're going to transition now to the lightning talks, and Jim DiCaprio is going to take over the podium.
Thank you, Tyler. Yeah, I'm Jim DiCaprio, physician scientist at Dana-Farber, and with Tyler, co-leader of the Bridge Project. And I wanted actually to welcome, there's about 40 Dana-Farber Harvard cancer investigators that are here in the audience today, responded. And for a physician scientist, hear these talks, it was just spectacular. I've stared at many PET scans and TC scans, and I certainly never imagined there was all that data that could be actually used so brilliantly, and seeing things that I never saw before. I mean, and just to come here today, even your AV system works, which is not something that works so commonly at Dana-Farber Harvard's cancer centers. So we're going to have a series of 3 lightning talks, and to highlight some actually currently funded bridge projects. And then this will be followed by a networking event, actually, where hopefully you can be inspired actually for applying for our next round of bridge funding, which will happen in early fall. So our first our speakers are Sahil Patel and Sagita Bhatia of the Koch Institute, and Leisha, again, from Mass General, to present on their work of a footbridge project from 2022 exploring alveolar macrophages as vehicles for collecting tumor DNA to improve diagnosis. Super.
Thank you for the opportunity to speak today. So as Leisha had mentioned, lung cancer is the leading cause of cancer-related mortality in both men and women. And as a result, there's been a big focus on trying to detect lung cancers early when they may be surgically curative or more amenable to treatment. And so we're catching, as part of the initial part of diagnosis, we have to identify pulmonary nodules, and we're catching millions of these every single year. And then the big question is a pulmonary nodule cancer or not cancer? And that could be really tricky for many of these nodules, and it's really hard to tell. And because of the risk benefits of biopsy, oftentimes we don't end up doing a biopsy because the yield may be too low or the risk may be too high. And so standard of care is essentially to follow them over imaging. And so we'll continue to follow them over time and see if they grow. And if they grow, then that changes our calculus in terms of thinking that this may be cancer. And when we think about that at its core, that whole approach is based on if there's a possible cancer, we're going to let it potentially grow as our test to see whether or not this may be cancer. And that seems backwards. And so we think that there must be a better way to better address this. Liquid biopsy is a diagnostic tool that we've been using for checking whether or not there's evidence of DNA from tumors that we can measure in the blood. And the problem for liquid biopsy right now with early stage lung cancers is very little, if any, tumor DNA ultimately ends up in the systemic circulation. And instead, most of that tumor DNA is cleared by tissue resident macrophages in the local environment. One of the things that we do as part of our procedures, with even as part of lung cancer workup, is something known as a bronchoscopy with bronchoalveolar lavage. And a bronchoalveolar lavage involves basically instilling some salt water into the area of the lung that we are trying to isolate. And we collect back samples from the local environment. And in the process, we're able to collect millions of alveolar macrophages, these same cells that are clearing a lot of the debris. And the benefit of a bronchoalveolar lavage is that we don't have to stick a needle accurately into a moving small target that we may not even be able to see. And so this led to our hypothesis that if we're able to collect from the local environment and collect these alveolar macrophages that are clearing the debris, can we leverage them as collectors of tumor DNA? And can we extract that DNA for the purposes of developing an even better test for lung cancer? And so to be able to do this, we had to bring together a team of different experts. And so we needed expertise in terms of sequencing, which we had Victor for. We needed an expert in translational bioengineering with Sangeeta. We needed a clinical oncologist who really understands early detection for lung cancer with Leisha Sequist. And my role is I'm a pulmonologist at Mass General. I work with Leisha in the pulmonary nodule clinic. And then I'm co-advised by Victor and Sangeeta in my postdoctoral research. And so to first study this, we started with the animal models, and we're able to create a mouse model of lung cancer. And we're able to measure plasma, as well as we're able to do a bronchoalveolar lavage for these mice. And we can look at the fluid from the bronchoalveolar lavage, and we can also look at the cells that we recover from that. And if we take the cells and we isolate the cytoplasmic fraction, which is where we would expect to see ingested tumor DNA, we can measure how much DNA we're able to recover from those. And we found that we're able to get a lot more DNA that we're getting from our BALF compartments, and we're able to get a lot of tumor DNA, which is what we're hoping for from our BALF compartments, both the supernatant and the cytoplasmic fraction, with 18 times as much tumor DNA in the BALF supernatant as compared to plasma, which is what we normally use for liquid biopsy, using our most sensitive test. And the cytoplasmic fraction has the potential for even more. But it's a race against time because the cells are basically degrading the DNA once they eat it. And so we want to think about how can we slow down that process. And so we started first with trying to genetically engineer some macrophages to see if we can prevent the degradation within the cells. And so on this first figure on the left, we have macrophages that are in the video showing a yellowish color, and they're eating dead tumor cells that have their DNA stained with blue-white dye. And essentially we see that they're picking it up and then they go away over a short period of time. And these images and video is taken over 30 minutes. But when we genetically modify the macrophages, we see that they accumulate. And so this was a great proof of principle for what we could do by slowing down that process, but genetically altering cells in humans isn't a very viable translational solution. And so we looked next at chemically to see whether or not we could achieve the same effect. And so we looked at small molecule drug inhibitors that are available that may have a similar effect, and found that with treatment with chloroquine, bifilomycin, and lyso-5, that we're able to achieve a similar result. And we're able to get more tumor DNA from our macrophages in cell culture. And chloroquine is an FDA-approved medication, which is something that is very exciting. And through this process, we're able to get 5 times as much tumor DNA recovery as compared to control. But as we look forward, we're looking to see a translatable solution that will help with diagnosis for humans. And what we envision is if we give a DNA degradation inhibitor before a bronchoscopy, and we let that accumulate, then we may be able to get more tumor DNA recovery. And first, to be able to assess if this is even possible, we have to understand what the baseline amounts are. And so we started by looking at human clinical samples and have an IRB approved and have enrolled 7 participants and have collected samples to get a baseline of how much is even there, and use this as a springboard to see how we can develop this proof of concept work into a translatable solution. And so I wanted to thank Sangeeta, Victor, and Lisha for their mentorship in this project, and really thank the Bridge the Bridge Project for allowing this kind of work to happen, because without their support, we wouldn't be able to make strides for this. Thank you.
We're gonna move on. So we're gonna hold off questions. Feel free to reach out directly to Sahil during our break. So we'll have our next speakers. Michael Birnbaum of the Koch Institute and Marcella Maus of MGH will talk about their traditional award. Bridge Project was awarded in 2023.
All right, good morning, everybody. It's a pleasure to be here. Marcella and I are excited to tell you about work that we have in process, combining some of the engineering approaches in my lab, in an immune engineering lab, with some of the clinical impact in Marcella's. So our project focuses on CAR T cells and CAR T receptors. So these are, of course, therapies which are transforming the landscape of care of certain types of cancers, particularly currently those for heme malignancies. And something to be noted is a couple of things. One is that there's a couple of different designs which are clinically approved, which achieve clinical efficacy in different ways. So in terms of the kinetics of cell expansion and cell persistence, in terms of the program that's given to the cells, there's actually a fair bit of differentiation between CARs expressing, for example, the CD28 costimulatory receptor versus the 4-1BB. So this is evidence that there's room to be able to have some type of real diversification of function. But into the clinic so far, it's still relatively limited, sort of the combinations of signaling inputs that have been tested. So if you sort of look, there's a few that are here on this list. But if you sort of just even take kind of a naive look in terms of, what would be in the back of an immunology textbook for what could be interesting immune inputs, the list is actually significantly larger. And so our bridge project is looking for a way to really adapt protocols in order to really take a much more expansive look at what would make a good CAR.
Thanks, Michael. So I'm Marcella Moss, and we are really happy to have assembled this bridge project. We each brought in trainees as well to help them develop their careers. Diego Salas Benito is a physician scientist from Spain who's in my lab, and Andrea Garmela is an MD PhD student here in the Harvard HST program who's being co-mentored by Michael and I to work on this project. So we really had two aims here. The first was to make a second generation mesothelin targeted CAR pool library building on work that Michael had performed before, but switching over to solid tumor and identifying second generation CARs, which is the kind of CAR T that's approved in clinic where there's just two signaling domains inside. And so we would need to build a new library to be able to do that. And so overall, we would generate about 6,000 CAR variants. We also decided in the course of the project that we would use two binder domains to two different antigens because that is an important aspect for solid tumors to overcome the tumor heterogeneity that's present in almost all patients. And so we would test a variety of hinge domains and multiple different co-stimulatory molecules from a library of about 20, along with 20 primary activating molecules called ITAM domains, and so build that library. And then our short-term goal was really to evaluate and validate the top hits of these novel CAR T cells in both a pooled format and a singular format using xenograft models that have been developed in our lab. So we use human T cells, transduce them with a library construct, and inject those cells into mice that bearing tumor that has been injected a week before. We can then collect that tumor, harvest the T cells, and then use sequencing analysis to identify which signaling domains really increased CAR T function and had increased persistence and infiltration into the tumor versus those that were selected against. And our long-term goal is really to be able to do this directly in patients, to perform IND-enabling studies in a phase 1 clinical trial of a CAR pool type of analysis of second-generation CAR T cells for patients with solid tumor. So the first part that I was going to focus on is identifying what the best antigen binding domain is going to be. And so we've been working on the best kind of binder in parallel with the best kind of signaling domain. And so Diego and Andrea have been working together to identify these two antigen targets for ovarian cancer. One targets mesothelin and the other targets MUC16, both of which are glycoproteins on the surface of ovarian cancer cells. And we made a series of constructs that have different spacer domains in between the two antigen binders. And we first tested them for just the ability to bind both antigens. And what we found is that one of our first ones called TAN-CAR1 and TAN-CAR3 are the ones that actually bind to both mesothelin and to MUC16. Now, I won't go through all of the various constructs that we made, but the key is that it's important to have the right spacing between the two to get optimal binding of each antigen. We then tested this in mouse models head-to-head, and we could see that, in fact, this TAN-CAR1, which is a particular configuration of binding mesothelin and MUC16, was able to extend the survival of mice, have the reduced tumor growth in the subcutaneous model after a single injection of CAR T cells given to these mice. And so in parallel, we've also been building a co-stimulatory library on that tandem CAR. This is from a range of different co-stimulatory molecules and activating molecules. We also built a library containing just one activating molecule and then a cytokine signal. So far, this one on the left seems to be working better. The ones in bold have been tested in some form of clinical trial before, but you can see there are many that are not in bold that have potential use in patients and potentially could increase the function of CAR T cells. So in summary, we really think that we need innovation in CAR treatments to be able to have clinical impact in solid tumors, such as ovarian cancer and other metastatic tumors. We've developed new CARs targeting both antigens, mesothelin and MUC16, for ovarian and other peritoneal cancers. And we really are thankful and grateful for the Bridge project to enable this convergence of both engineering and clinical expertise. Thank you.
Really spectacular, thank you. So our last talk before the break Louis DeRitter and Doug Rubinson of the Dana-Farber will speak. Giovanna Traversa and Bob Langer unfortunately couldn't make it today. But you know that Bob, of course, is totally interested in the bridge project and already emailed me today about a new idea he has for our next funded. So.
Could you please project the slides up? All right, thank you. So it is a pleasure to present on behalf of the full team here. I was very lucky very early on my PhD training with Bob Langer and Gio Traverso to start collaborating with Doug Rubinson on this work. So very excited to share this on behalf of the full team. So if LeBron James and Happy the Dwarf were both to present to the clinic and they both had to get a prescription of the common chemotherapy 5-fluorouracil, like multiple different chemotherapies, they'd get the same dose of chemotherapy. And the reason for that is although they have different heights and weights, they have the same ideal body surface area. And what's even more incredible is the way that that dose is determined, that is based on that body surface area, is calculated from this equation from 1916 that was empirically fit to data from just 9 patients. And in 2024, in the top cancer centers in the world, like the Dana-Farber, this is still how it's done. And as you're probably thinking, that doesn't make any sense. There's multiple different things that are different between LeBron James and Happy the Dwarf that are not captured in those body surface area measurements. And those can be both at the interpatient level, so people with the same body surface area, they could have very different responses to the drug. And also within the same patient over the course of time, whether it's the same round of chemotherapy or one round of chemotherapy to the next. So to do what we love to do here at MIT is solve these problems. We went ahead and developed this system to make it so that you can personalize the dose of the chemotherapy to the given patient. And to do that, we developed this closed loop system to make it so that you can personalize the dose to the patient. So a closed loop system, they're used in everyday life like a thermostat, and we just wanted to apply that to the clinic. So in the closed loop system we developed, what we do is we take your desired concentration of the drug, in this case 5-fluorouracil, and compare that to the measured concentration of the drug. And that difference is the error. That goes into this control algorithm, which then adjusts the infusion rate into the patient, and that gives you the actual concentration of the drug. But in order to do the closed loop sensing system, you have to measure the drug concentration. So to do that, we sample blood from the patient, perform this rapid blood pre-processing step. And that clean sample then goes onto our sensing system that gives you the measured concentration of drug. And it's very important to note that our sensing system that we use here is HPLC-MS, which is widely used and trusted by the FDA to determine drug concentrations throughout clinical studies. So by using that as our sensing system, we de-risk that part of the overall system. And we really designed this entire closed loop system around the LC-MS for that reason. And we refer to this system as Claudia, which stands for the closed loop automated drug infusion regulator. So you'll hear me refer to Claudia, and that's just this overall system. So the question then is does this system work? Can we actually control the concentration of drug, in this case 5-fluorouracil, better than the standard of care of body surface area dosing? So in a study of animal experiments, we gave a constant rate infusion on a BSA basis to rabbits. And shown here in the shaded blue region is 2.5 to 3 milligrams on the liter, which is the commonly accepted target therapeutic window for 5-fluorouracil, which through some studies have shown that it has its best efficacy, and time shown on the x-axis. So you can see here that if you guess the dose correctly, you can end up within the therapeutic window for some of the time. What's important to note is we use the same exact dose of 5-fluorouracil in this study shown right here. And what you see on the y-axis is the concentration range is different. Here I have the max of 6 on the axis, here's 20, and that target window is shown here in shaded blue. And what's very important to point out is in the clinic, it's very well known that 5-fluorouracil experiences 8 to 10-fold pharmacokinetic variability, and that's what we see here. So we've been talking about it amorphously, but concretely, this is what the clinical reality looks like for people getting 5-fluorouracil and multiple additional chemotherapies. And as you're probably guessing, our closed loop system, Claudia, is able to control the concentration under both those different conditions. So this system does indeed seem to be able to control the concentration of 5-fluorouracil over clinically relevant conditions. And I'll let Doug talk about his thoughts of this from a clinical perspective.
Excellent. Thank you, Louis. And thank you for inviting me. It's great to be back here. I finished my PhD a few years ago here, and what an awesome day. Clinically, I want to make the unsexy sexy. We all read the New England Journal. We see these KRAS inhibitors, amazing drugs. They add months to our patients' lives. A day of a KRAS G12C inhibitor is just under $1,000. We're going to extend your life by 4 or 5 months on average. It's wonderful. 5-fluorouracil has been in use since the 1970s. It is the most important drug in GI cancer. It is the only drug in combination that can cure someone after surgery for colon cancer, pancreatic cancer. It can add not days or weeks or months, but years to patients' lives with colon cancer, with incurable disease. And we're using it wrong. We're using it wrong because we've known for a long time now that we're underdosing a lot of people. They break it down more quickly. We're not getting to the concentration needed to kill their cancer. We're using it wrong, and that we're overdosing people, and people are coming in with severe side effects, and we're needing to adjust. Well, now we have the technology to start using it correctly to get people the dose of the chemotherapy to treat their cancer most effectively, cure hopefully more people, extend people's lives, avoid toxicity. This is really exciting to work with great collaborators in Gio Traverso, Bob Langer, Louis, to figure out how we can get this into our patients' hands. Thank you.
Thank you, Doug. And just to close, is one of the real key advantages of using HPLC-MS as our sensor is it's very easy to apply to additional chemotherapies. So although we focused on 5-fluorouracil here, we see that we could easily adopt very trivially in about a week of work the system to control additional chemotherapies as well as multiple other drug classes. We really think that any drug that could be measured, its concentration by LCMS, which is the vast majority of drugs, could be controlled by the system that are given by infusion. And just some ideas of some additional drug classes that may benefit include immunosuppressive drugs, antibiotics, and even perhaps anesthesia. So with that, I'd like to say thank you to a huge team. It was a huge team effort to make all of this work happen. So I want to be sure to thank all of them. And I'd be very remiss to not thank the Bridge program, which has really helped us continue to develop this system and hopefully be able to impact many patients' lives. So thank you all.
Thank you very much, Jim.
Thank you.
Terrific. Well, that was a spectacular morning. You know, the Bridge Project is trying to bring these MIT engineers that can measure like vastly small and incredibly complex data sets with physicians and scientists in the Dana-Farber Harvard Cancer Center who face the real world of cancer in all its varieties. The Dana-Farber Harvard Cancer Center has investigators from Children's Hospital, Mass General, Brigham, BI, the Public Health School, the Medical School, and Dana-Farber. And we're facing this enormous challenge. So the Bridge Project is really a first step in trying to make this happen and bring some brilliant people, put them together. As Art Gelb actually told me, he says, you know, MIT, we can measure stuff and we can help this cancer problem in a way that is really awe-inspiring. So I want to thank the Gelb family for actually starting this whole thing, the Goodwin family for actually really making it such a big project as it is now. And I want to thank you for all your attention. We have a networking event now. So I'm going to ask you all to leave the room because the MIT engineers are going to rearrange this room actually to a networking event. And so just in a few minutes, but there's lunch outside for everybody, who, especially those who've pre-registered. And then we'll have a networking event organized by themes, and we have posters. And to the young people, it's not your time to be shy, okay? Reach out and ask questions and make some new friends. Thank you all.